Seftel, Research Director for the Macular Degeneration Foundation, and we're here with Professor Ed Boyden from MIT. Welcome, Professor.
Thank you. It's great to be here.
Tell us a little bit about the presentation that you're going to be making at ARVO 2017.
My group at MIT develops technologies to help us understand and treat diseases involving the nervous system. So to analyze the brain, we have to figure out how to map the molecules in the cells. Right. And we found a really great new way of doing it. Yeah. So I'll talk about the technology, expansion microscopy, that we've developed. The flip side is how to treat diseases. So we've been developing ways to control brain cells and neurons and cells of the eye using light. And so we have certain molecules now that convert different colors of light to electrical signals, and we hope that we can help restore, amongst other things, vision in the case of RP, macular degeneration, and other diseases involving retinal changes or loss.
What have you identified as the central weakness in macular degeneration in terms of neurotransmissivity?
So my group is an invention group. We develop tools, and then we try to distribute them throughout the world to help people answer questions about how to fix problems of the retina and of the brain and of the rest of the body. In the case of retinal blindness, one of the technologies we've been developing are so-called optogenetic tools. These are molecules that convert light into electrical signals, and they're encoded for by small genes. So one of our hopes is, in the case of conditions like RP and macular degeneration where photoreceptor cells have atrophied, can we help people deliver these molecules to different levels of the retina that have been more spared than the photoreceptors, retinal ganglion cells, bipolar cells, and so forth, in order to convert them essentially into a bit of a camera, so they can capture pictures of the world and transmit that information to the brain better. At this conference, we also talked about ways of making these molecules very localized. That helps increase the resolution of the camera, if you will. If the molecules are everywhere, then the image is a bit blurry because the cells can capture light from all over the place. If each cell only has one area of the cell that's photosensitive, though, it's like having a high-resolution camera.
If we look at the area of stem cell therapy, one of the central challenges is you're introducing stem cells that are not connected to the existing retinal electrical infrastructure. Is there a role for the work that you're doing in helping to re-innovate or reconnect stem cells that are implanted into the subretinal space, for example?
There are two really important applications of our tools to such stem cell applications. One is, how do we know what the stem cells should look like in the mature state? You know, when we do regenerative medicine, it's often a bit open loop. We try to turn a cell into a target cell, but it's hard to then to characterize that target cell. Using the method I mentioned where we called expansion microscopy, we hope to actually map out what the circuit should look like. That becomes our goal, and then we can coax stem cells into that final shape and do sort of closed-loop tissue engineering. Can we actually try a different cocktail of genes or small molecules, turn a cell into a target cell? It matches in some ways, but not others. Use the map to correct, and over a couple cycles of iteration, can we actually converge upon the target? The second is the optogenetic tools, where we control cells with light. Can we drive cells actively and use that plasticity to alter the wiring? You know, because it's important not just to have the cells there, they have to wire up in the right pattern. And arguably a lot of the challenges of getting stem cells to integrate into neural circuitry must be addressed by either delivering molecules to recapitulate certain developmental processes or to use activity to cause the wiring to be in the correct pattern over the history of development and rejuvenation.
Two approaches, rather. Which of those do you believe is closest to clinical application?
Well, I really am excited about the idea of closed-loop tissue engineering, where we have a map and we use that to guide our protocols to turn cells into their target cells. I actually help set up a center now called the Center for Genomically Engineered Organs, along with George Church, Jennifer Lewis, and Ting Wu across Harvard and MIT. And the goal is to implement that closed-loop mapping. We're trying to develop ways to take a cell, differentiate it, and then map out how all the genes are expressed in that cell and its shape. So I think that in the coming years, my hope is that we can actually map out the molecules that are required to turn a cell into a target cell. At that point, we could really hopefully turn stem cell engineering into a design science. Can we actually say, look, do these 17 things and you're guaranteed to get the cell you want? And arguably those roadmaps are still emerging. So there's some fundamental science to be done, it seems to me, in order to make sure that our recipes for making cells what we want, and for example, not having them turn into tumor cells or other byproduct cells, to be very rigorous and accurate.
Right. So what are your goals over the next year for your current projects in this regard?
I'm really excited about the idea of mapping life. Can we actually try to map out the molecules throughout cells and the cells throughout tissues, both to give us goals for regenerative medicine, but also to pinpoint disease targets in the disease state? To do that, we're developing a technology we call expansion microscopy, where we take a tissue sample, we synthesize throughout the cells, throughout the spaces between the cells, around the molecules and between the molecules, essentially a three-dimensional spider web of swellable polymer. We can add water, the polymer will absorb the water and swell, pushing all the molecules apart until they're far enough apart we can actually tell them apart. So could you actually map all the molecules in a cell and the cells throughout a tissue? My hope is to make that a reality in the coming years, and then to launch an atlasing project where we could try to map out all of the molecular changes that occur in disease. My hope is that will give us very precise drug targets for disease and also give us guidelines for how to do regenerative medicine towards very specific goals.
When you do this mapping, how do you transcribe the map? Have you developed a software application that allows you to visualize this in 3D, for example?
Up till now, we've mostly focused on the chemistry, but now the chemistries are getting good enough that we actually think it's time to really focus on the hardware and the software. I'm part of an international collaboration sponsored by Cancer Research UK to try to make a virtual reality map of breast cancer. And the basic idea is use our expansion method to blow up a tumor, map it using a microscope, and then visualize it in 3D using virtual reality. But why can't we do that for all diseases? It seems like a common problem with all modern diseases is there are three-dimensional problems of large-scale organ systems, but the pathology often begins with nanoscale changes in genes or proteins or parts of cells. And so I think this tension of visualization applies to all of these modern diseases. It's not just take an antibiotic and wipe out the bacterium. These are three-dimensional problems. So in the next couple of years, I hope to build more coalitions to bring together the visualization along with the hardware. We're talking to lots of people about building very high-speed, very cheap microscopes to acquire the data and to fuse that with our chemical tools for visualizing diseases and disease states with incredible accuracy.
So what you're talking about is developing a virtual human, an interactive virtual human model that scales from nanoscale to macro scale.
That's right. I think if we can find a way to map out, because here's why I think this is so important. If you look at a lot of modern diseases, metabolic diseases, cancers, diseases of the nervous system and of the eye and of the ear, autoimmune diseases and so forth, they all have this common pattern that they involve large-scale three-dimensional body systems, but the pathology inevitably begins with these nanoscale changes, proteins and biomolecules and so forth. And so I feel that one of the biggest needs in modern biology and medicine is the ability to visualize what happens across these scales.
Right. Because as clinicians, we tend to look at disease in a very linear and a sort of an additive fashion, rather than looking at it as a dynamic system where a lot of these equations are logarithmic. A lot of these equations have different, have complex mathematics associated with them that is not captured by our very binary and simplistic current analysis. So what you're talking about is giving a new set of tools that add not only granularity, but also a level of the sophistication of some of these interactions.
Well, I think that's important, not just because it will give us really great data, but because the data will inspire maybe even less invasive, more powerful therapeutics. I'll give you an example. One of my colleagues at MIT, Li-Wei Tsai, recently led a study that we helped with, where she used our older tools, the optogenetic tools, where we activate neurons with light, to discover that certain patterns of brain activity might actually help clean up some of the molecular problems associated with Alzheimer's disease. From that insight, our collaborative teams then put together an idea, which is why can't we deliver visual stimuli, synthesize the same pattern of brain activity through just our senses, and then help correct Alzheimer's. And Li-Wei and I just co-founded a company, Therapeutics, to try to actually do human clinical trials on this idea. Imagine a movie that would cure Alzheimer's disease. So that discovery, though, would have never been made without the insights about the patterns of activity in the brain, and those patterns would not have been revealed without our older tools for controlling neurons with light. So mapping, whether it's watching or perturbing, and then collecting that data, can inspire potentially therapies that are very powerful and very non-invasive, because we've understood the principles of the disease.
Right.
And my hope is to get a couple more success stories like this, as part of the education, but also to help cure people, in order to inspire whether we can actually take precision medicine and make it as real a thing as we can, and also to understand the principles that lead to diseases in order to see if we could just tweak the control knobs a little bit and steer the system back to a more healthy state.
I think it's very interesting because over 50 years ago, the Russians were doing experiments with microcurrent stimulation of the periocular area. And they reported some data saying improvements in macular degeneration simply by stimulating, electrical stimulation across the retinal membrane.
Yeah.
Nobody's really explored the science behind that.
That's right. Well, the good thing about these kinds of therapeutic possibilities is that they could be very non-invasive. What often is necessary to make it into a true engineerable therapy is some insight into mechanism, right? What is the electricity going to do that's positive? Could there be side effects? And then to understand the molecular cascades that are engaged. In the collaboration with Leeway on Alzheimer's, they actually found that this particular pattern of neural activity caused the immune cells of the brain, the microglial cells, to go into a more healthy state. And they would then start cleaning up the debris nearby. And at the meeting already, I've had some chats with people about the microglia and other immune cells of the eye. And could there be some commonalities? Are there patterns of activity that could be linked to healthy molecular cascades? And if you can engage them through stimulation, through non-invasive mechanisms, could that potentially be a way of repairing the eye without having to inject factors or other kinds of invasive manipulations?
To summarize then, you've given us a sense of what you're going to be doing in the next year. What is your vision for the next 5 years?
So my hope is in the next 5 years, we will have completed some maps of disease states versus normal states. We'll have some ideas about novel clinical targets, which were not predicted by hypothesis, but have to be mined from new data. And maybe we'll have some new insights into therapeutics, like the Alzheimer's intervention that I mentioned, which now we're starting human clinical trials on. I also hope that we'll have better technologies for non-invasive monitoring or minimally invasive monitoring that give us molecular information and also a great deal of anatomical insight.
Right. Excellent. Professor Boyden, thank you so much for spending this time with us.
Great.
Really appreciate it.
Really good talking to you.
Thank you again. Yeah.